One moment, Rich, we are having some, let's see if this unmuting. We can't hear you.
I hadn't said anything yet. Anyway, it's going to rain forever. We're in a rainy phase, which is really sad. The weekend was beautiful, except Monday was a little hot, but the other 2 days were spectacular. So we're paying for it now with like 10 days of rain. Might need to get the arc out. This is Migraine and Headache Awareness Month. Will put out a little bit of some history of migraine, first described in 3000 BC, with pain so severe the patient wishes for death to escape. Then he describes the description of botulism, the development of triptans in 1991 when I was sitting in your seats. In 2010, the FDA finally approved botulism for the treatment of migraine, long after it had approved it for dystonias. Today is Dr. Klotman's State of the School address. It is at 4 p.m. in the Great Hall or via Zoom. She's going to honor a surprise member of the Department of Neurology. Anyone know who that is? No? Will, do you know?
I don't. I asked, but they wouldn't tell me.
Well, presumably that person's been invited to be there, so they know. Again, the race against ALS, which is coming up next weekend. Matt, Don Sanders, and Mark Skeen are all racing in their supercars. If you want to donate $100, you get a chance to pick which one you want to race with, or at least do a lap with. You can scan this QR code. A lot of going on the research front. Katie Peters was part of an article on a new treatment for low-grade gliomas in the New England Journal of Medicine. Chris, Elijah, Suma, Kristen looked at complications of hydrocephalus in patients with neurosarcoidosis, all of whom had a normal ACE level, I would say. Ryan McCrory, Cheryl Shree were part of a study in JAMA that looked at vitamin K antagonists that did not appear to increase the risk of complications from thrombectomy. And Atif was a co-author of a statement on qualifications for neurodiagnostic personnel in muscle and nerve. And Jordan Mayberry was in Forbes Health. Isn't Forbes magazine bankrupt? Yeah, they were just bought for almost nothing by some company. Some rich person just bought it. So it's no longer owned by the Forbes. He talked about the symptoms of neuropathy. Why does Katie Moore love being a neurologist? First, she likes the neurologic exam, which I think drew a lot of us to this field. And then she talked about how she enjoyed the partnership with patients over years, which is part of being a doctor who sees people with chronic diseases. So thank you for that, Katie. Some upcoming events. So we have a special faculty meeting on finances and DHIP, describing contracts and stuff next Wednesday at 5:30. It'll be Zoom. The end of the year faculty meeting, which used to be JT's meeting, is August 7th at 5:30. We're still not sure if we'll have it here or in the Bryant building. The theme is going to be the 10th year anniversary of the department. The new resident and fellows welcome event is July 13th at Duke Gardens at 6, and the annual Durham Bull game is September 1. So a lot of events coming up. I'm putting together the Grand Round schedule. So anyone who is interested in talking before I reach out to you or wants to recommend somebody either at Duke or otherwise just send me an email at neurochair, which is my secret email, or you can email Tico. Just a little bit about North Carolina, which, as you know, I'm quite fond of. The oldest cities. See how many of you know the oldest city? They even know which is the oldest city in North Carolina? Okay, so just by example, Charlotte, 1768, Raleigh, 1792, Durham, 1853. So the oldest ones all started on the coast. The sixth oldest was Hillsborough, which is in the middle, 1754. It's also home of the world famous wooden nickel. Brunswick, 1725. It doesn't really exist anymore because the British burned it to the ground in 1776. Beaufort, 1730. We're getting older and older. That is where the Duke University Marine Lab is. It's that little yellow spot there. Edenton, 1712, was site of the Edenton Tea Party, a protest organized to support the Boston Tea Party. Number 2 is Newbern, which is the green dot, 1710, named after Bern, Switzerland, the capital city. It was North Carolina's capital for over 20 years. All right, nobody knows? I didn't know until I did this. So the oldest city is Bath, North Carolina, again on the coast, 1705. It was the site of Carey's Rebellion, and it served as the base for Blackbeard the pirate. When I saw it was the site of Carey's Rebellion, I thought to myself, is that the same Carey who founded our Carey, south of Raleigh? And it turned out it wasn't. So our Carey was founded by Allison Francis Frank Page. He was the developer, mayor, postmaster, and railroad agent. And his wife, Kate, bought 300 acres. And he named the area Carey after Samuel Fenton Carey, a prohibition leader from Ohio. And you now know there's a big development in Carey called Fenton. So pretty soon there'll be something called Samuel there as well. So that's a little history of the state. And again, today's CME code and our case presentation is Amber Anderson. So Amber, I'll turn it over to you. Stop share. Get rid of my slides. So this one? On the internet, okay. Here, you see through it.
Okay.
You got to share screen.
Oh. Hang on.
You got to share screen.
Okay.
So,
on that.
Yep. Good. Yeah. And you can get rid of that by hide floating control.
All right. Well, good morning, everyone. And on Zoom, my name is Amber. My case today is a 69-year-old male with a past medical history of AFib, complete heart block. He has a pacemaker, dilated cardiomyopathy, who presented to the Duke Neuromuscular Clinic with chronic bilateral arm weakness and gait instability. He was actually referred by his cardiologist who noticed the worsening arm weakness. He reported this for around 5 to 10 years and had just gradually worsened. It now became difficult for him to perform his normal activities like lift a gallon of milk or climb up stairs due to fatigue. He also reported trouble with his right hand not wanting to release and kind of described this as like a Charlie horse feeling. And he had some gait instability for around 2 years without falls. And he notable history of myotonic dystrophy in his father. There we go. So his exam, he was alert and oriented. Cranial nerves were notable for some facial weakness. So he had moderate cheek puff weakness, moderate eyelid weakness with Bell's phenomenon. He had lingual myotonia with his tongue protruded midline. And motor-wise, he had some decreased bulk in bilateral pec muscles, some mild right scapular winging, bitemporal atrophy, and right bicep fasciculations. And strength-wise, he had really weak grip and deep finger flexors, and also proximal weakness in upper and lower. And he had some thenar and forearm percussion myotonia, right greater than the left, and some grit myotonia, right greater than the left. 1+ reflexes throughout, decreased temperature on the left foot, intact coordination. And I tried to find a video of the closest gait I could find. Hopefully it plays. Oh. Well. Yeah, it worked earlier. I think it worked if I can get out of this and then go there. Okay. So you'll see here, this is a little boy. He has a lumbar hyperlordosis. His shoulders are kind of thrusted back. His abdomen is very protuberant. This places his center of gravity kind of behind his hips so he doesn't fall forward because of his weak back muscles and weak hip extensors. So this is how my patient was ambulating. So the workup for this gentleman, since he came in with a positive family history of myotonic dystrophy and kind of a classical presentation, we decided to get genetic workup for type 1 and type 2. So type 1 is the DMPK gene and type 2 is the CNBP gene. And he came back positive for myotonic dystrophy type 1. So what this is, it's an autosomal dominant disorder. It's a CTG trinucleotide repeat expansion of over 50 repeats on the DMPK gene on chromosome 19. And the diagnosis is consistent with over 50 repeats. So you can see here he had over 200. So what is myotonic dystrophy? So myotonic just means inability of the muscles to relax at rest. And then dystrophy is the degeneration of the muscle leading to weakness and atrophy. And again, autosomal dominant has a very strong genetic anticipation. It's very variable. The most common onset is adult in the second to fourth decade of life. And they have weakness in their distal muscles, some atrophy, bilateral ptosis, and myotonia. And this is a multi-system disease process. So the top three, cardiac involvement, so they can have cardiomyopathy, they can have complete heart blocks, ventricular tachycardias, bradyarrhythmias, orthostatic hypotension. So it's always a good idea to obtain a baseline ECG while you're in clinic. And other neurological things I haven't mentioned is they can have an apathetic demeanor, also have some learning disabilities. They can have some daytime somnolence, greater risk of developing obstructive sleep apnea. And pulmonary, this is unfortunately the leading cause of death in these patients because the diaphragm muscle is involved, can cause some weakness and myotonia, lead to respiratory distress, and in severe cases can lead to mechanical ventilation. And they are also at high risk for aspiration. So you can get an FVC while you're in clinic with these patients, supine and upright, and make sure they're up to date on their vaccinations like flu and pneumonia. And also they can get a cough assist device if needed. And again, these symptoms are very gradual, and cataracts are also very common with these patients. And you can see here on the right, these are not my people, but seen on the very top is bilateral. He has bilateral ptosis. They have bitemporal atrophy. Their masseters are atrophied, very long faces, frontal balding. And then the diagram over here highlighted in red, the dark red is all of the muscles that are involved. And last but not least, these patients are a very high rate of complications and even death during given with anesthesia during surgery. So you always want to tell them if they need surgery to tell their surgical team to be aware that they have myotonic dystrophy. And also acetylcholine should be avoided because of the potential to cause diffuse muscle contractions. And that's it.
So while he's getting his PowerPoint ready, I'll go ahead and introduce. So welcome again this morning for our Chief Grand Round series. We just have this week and one more week for the year, and then everyone will be headed off to fellowship. So today we're going to be hearing from Jung Ko. So Jung actually is a former Duke undergrad student who we persuaded to come back after he absconded to UNC for his MPH and MD. He's been with us obviously for the last 4 years, and he's liked it so much since he came back, he's going to hang on for another year of fellowship in neurophysiology. And then after that, I think he's still looking for a place to land, but he's been interviewing around, and I'm sure he'll find a place here soon.
I hope so.
So I actually don't remember the topic is because he's pulling it up.
Yep. Amber, do you have the link to that box there? Yeah. So let me just go to box. All right, so that was a lot of clicking under pressure there. Okay. Okay. All right. Thank you for your introduction, Dr. Eckstein. For my grand rounds topic this morning, I want to talk about drug development and future directions. And sorry for the delay again. No disclosures. So let's start with why I picked this topic to start with. Looking back on my medical education of 4 years residency, 4 years medical school, and undergrad before, I think most of the focus has been on patient care and clinical reasoning, which is understandable. That stuff takes a lifetime to master. But I think looking back, there's a certain lack of understanding when it comes to the big machinery that is the healthcare space in the United States. And I think pharmaceutical industry plays a big part in that. And we're medicine doctors after all, we're not surgeons. Medicines are tools, primary tools. So we should know how the tools are made, not just how to use them. In academic centers like this, our role with our interface with the pharmaceutical development is often limited to phase 3 trials, but there's a lot of career opportunities available for those who are interested to be involved with any step of the development process. And I think most importantly, as we sort of progress through our careers and become more involved with patient care and hone our skills, learning more about this process can help us develop better informed prescribing habits based on safety, efficacy, and costs. So let's see. So roadmap for this talk, I'm going to first talk about the drug development process, very sort of a broad overview of it. Look at the pharmaceutical R&D landscape and how recent advances in machine learning and AI can help with this process. And I'm going to go over an example case of that. So I think it's always good to start with a definition. Let's start with what are drugs, the very basic question, right? So it's defined as biological or chemical entity that can modulate the course of disease by interacting with a certain protein by inhibition or activation. Makes sense to everybody. There's 2 broad categories of drugs. One are biologics, which are usually antibodies, and there are chemical entities, which are small molecules. So with that definition, let's go back about 150, 160 years in the past to Dr. Crum, Brown, and Fraser in 1868. So this was the time when they first described this idea of structure-activity relationship, where there's a relationship between chemical structure of a molecule and its biological activity. So if you change the chemical structure of a molecule, you're going to affect its biological activity. It makes sense to us now. It's common knowledge, but this was considered revolutionary back in the day, and it kicked off modern pharmacology. So this is Dr. Crum, Brown, and you can actually find this 1868 paper on PubMed. It's not like it's PDF scanned in. You can see its title is on the connection between chemical constitution and physiological action. And they note some of the compounds they studied there with old names. I think Codia is probably codeine, morphia, morphine, nicotia is nicotine. I had to look up what strychnia is. I don't know if anyone knows that one. Apparently it was used as like a nutritional supplement for pregnant women in 1800s. But nowadays, if you have a like mole or groundhog infestation, that's what you go for. So it's highly toxic. You don't want to do that anymore. So with this idea of structure-activity relationship, they started to extract pure chemicals from known medicinal plants. So morphine was the first to be extracted from opium. And another famous example is digoxin from digitalis for arrhythmia. And this is called forward pharmacology, where you extract active ingredients from medicinal plants without the knowledge of specific biological targets. So this was sort of first to be done. As we move forward in history, we develop our understanding of biochemistry, biological pathways, and we actually start to have some targets that we want to go after. So we learn about beta-2 adrenergic receptor and H2 receptor, and we screen for molecules that exist in the world that can actually bind to these molecules and affect the receptors and hopefully affect the disease state. So early examples are salmeterol for asthma and ranitidine for stomach ulcers, right? So this is called reverse pharmacology. So you're starting with certain target receptors and you're trying to screen for molecules that bind to those receptors. And it's also called target-based discovery. So this is a diagram showing sort of the difference between the two. Again, forward, you start with functional activity, active ingredient, and then find the targets later. Whereas reverse pharmacology, you have a target in mind, you screen for molecules, and then you come up with hopefully a drug that works. As you probably know, reverse pharmacology is what sort of driving the pharmaceutical research now. So I'll mainly focus on that. So if you Google drug discovery process on Google Images, you find tons of charts like this with different colors and sequences, but most of them follow the same sort of pattern. You know, from our side in Academic Medical Center, we're familiar with preclinical and clinical side of it, so the right half of the drug discovery process, but we may be a little less familiar with the sort of strategic research and exploratory research and candidate drug selection. So I'll talk about those. So exploratory research part. So that is the goal of that section is to find candidate molecules that will actually bind to the target molecule. So how do we do that? First method is to run a computer simulation. That's called in silico virtual screening. So you're looking at millions of candidate molecules and run simulations on your computer. And you try to find the target molecule that you can actually run experiments on. Next one is high throughput screening, or HTS. This is a technique that was developed decades ago, but sort of revolutionized the field of reverse pharmacology. So I'll talk about that here. So what happens is you have a microtiter plate with, you know, ranging from wells, 96 wells to 6,000 wells. And you can imagine these little wells are filled up with reagents and target molecules. And you have robot that can pipette really well and have into each well different concentration of candidate molecule or different candidate molecules into all these wells. I guess before this happened, we were just paying like medical students or undergrads to do this. But now we can do this. There are some systems that exist today that can test up to like 100,000 compounds a day. So very impressive. And this system can also run assays automatically. Often we try to have a threshold of some measure of interest. And if it's positive, you call that a hit. And if it's negative, you just screened out. Let's see here. So this is what it might look like. You have this fancy robot that's doing this for you. And then you have a microtiter plate filled up with target molecules and candidate molecules that you want to see how they react. So, even with robots, this is an expensive process. So you don't want to repeat things if you don't have to. So people started sort of combining their experimental data together in multiple databases. This is one of the more famous ones. It's called Campbell. This is something you can access online for free. I think it's maintained by European Molecular Biology Lab. And you can see it has currently 2.2 million compounds with, and they have a lot of reactions they recorded. And you can see a lot of different sources they pull from. So this is a molecule that you might see on their database. Can anyone guess what this is? This has 2 benzene rings and a diazepine ring. So it falls in the category of benzodiazepine. And this one is our favorite, lorazepam. So you can find this on the website, and you can see like 20 pages of chemical information on there. What might be of interest to us is the drug mechanism section. You can see it is a GABA A receptor anion channel, binds to GABA A receptor. It's been tested in people, and it's got references here. So say you have the top of the line screening tool where you can screen thousands and 100,000 compounds per day. And there's a range of hits that you get ranging from 0.1 to 10%. So you still are going to get about 1,000 hits each day. Now you can't take 1,000 molecules to preclinical and clinical trials, so you still need to pare that down. So this process of paring down hits to just a couple of lead molecules is called hits to leads. So how do we start to filter this 1,000 hits we get from the screening stage? You might say, we should probably just select for the most potent hits. Potency, I mean, it's just select the molecules that binds the best to the receptors. So one way to measure that is the inhibitory constant K. So this, if you remember from biochemistry, this is the concentration of a molecule at which 50% of the receptors are bound. So lower this number, less you need to bind to 50% to some more potency. So lower, more potent, right? So you might decide, I want to lower my threshold to maybe 100 nanomoles from 10 micromoles, and that will weed out a lot of less potent molecules. But the problem we run into with this tactic is the dynamic between pharmacokinetics and pharmacodynamics. Pharmacodynamics is only concerned with how strongly molecules bind to the receptor, but big part of drug development is pharmacokinetics, which is how our body processes the medications. Can we actually make use of this molecule as the receptors are bound? So just selecting for most potent hits ignores this second half and often leads to drug development failure. So you just don't want to do this by itself. Another thing to consider is the structural complexity. Oftentimes when molecules are going from hits to leads to clinical trials, you need to add on more molecular groups to make it more bioavailable or less toxic. And so if you already start off with complex molecules that are difficult to synthesize, this becomes much less attractive. So you want to in general, pick simpler molecules that does the job. So this guy Lipinski, I think he was a pharmacist back in the 90s, came up with this rule of 5, which is actually 4 rules. That's very annoying, where he looked at large set of over 2,000 orally active drugs and looked at their structures and try to see what are some characteristics of their structures that make them good oral drugs. And he came up with 4 different rules. So molecular weight being less than 500, lipophilicity less than 5, it should be more hydrophilic than lipophilic. And you see hydrogen bond donor, requirement, and hydrogen acceptor. So this rule can be applied to your 1,000 hits to hopefully pare it down to a more manageable level to the next step. But that's not the only thing you should consider. You need to also consider, is this toxic? Can it even be synthesized realistically? Is this metabolically stable? Can it be even absorbed? So there are a lot of factors to consider to sort of pare down. And you can see in the later slides why this, so many factors leads to a high failure rate of drug development. Fortunately, there have been a couple instances where pure dumb luck went into effect in discovering new drugs. So I'm just briefly touch on 2 examples. Penicillin is a famous example discovered in 1928 when Alexander Fleming went to vacation and came back and apparently left a little crack in the window and had a bunch of petri dishes with staph colonies laying around. He noticed that there was some contamination with the mold in a couple of petri dishes, and around that mold, no staph was growing. Right? It took about another 10 years to actually isolate this compound from the mold. Actually, I don't know if the mold was called penicillium to begin with, or is this after a drug, but doesn't really matter. Anyway, so 1939 to 1941, we start to experiment with penicillin. And 1941, we have actual human trials. And I guess that was expedited because US enters World War II and it starts being used then. Okay. Second one is Librium. This was discovered at Hoffman LaRoche tranquilizer development in 1957. They described this episode as just literally a compound that was developed 10 years ago that was sitting on a shelf in the lab. And they were kind of giving up on tranquilizer development and cleaning out the lab, and they found this vial. So they tested it, showed a high activity, low toxicity. This was the first benzodiazepine to be developed as a human drug. So you can see a lot of factors consider, a lot of investment in sort of lab and research. So the estimated cost of drug development runs from $300 million to $3 billion in the US with high failure rate, over 90%, and development of approval takes over 10 to 15 years. This is a table I got from GoodRx of the top 10 most expensive medications in the US now. I did not recognize most of this medication. I did hear of number one, which is costing over $2 million. That's a genetic therapy for SMA. And this is annual cost based on length of therapy. And who can really afford this, right? So what are some factors that's affecting drug pricing? I think the most important one, actually most important two factors are probably based on the indication. So if the population you're treating is, say it's folks with hypertension or hyperlipidemia, or it's millions and millions of people, you probably need to price the drug lower. You could get away with that and then recoup the benefit, recoup the investment you made in R&D. Whereas if you're developing drug for SMA, which affects thousands of people in the orders of that, you're going to need to raise that price higher to recoup the R&D costs. Other minor factors include accounting for rebates and discounts and sort of in comparison to what's on the market. But it's sort of a black box how they price these things, and but I think sort of working on all these factors could hopefully bring some of these prices down. And maybe this guy had something to do with it. Anyway, I think R&D decreasing the cost of this could potentially help with lowering the drug prices. So let's see how ML can help do that. So as we talked about, in vitro data are expensive, time-consuming, laborious, and computational models have been shown to quickly build a large set of compounds and predict the pharmacokinetics and bioactivity. So these are some of the questions that can be answered by computational models. These include like what target proteins could this bind, what other compound, what other protein could this compound bind to, how can the structure of this compound be modified to enhance pharmacokinetics bioactivity. These are all the questions that current sort of algorithms can answer. And before we go more in depth, I think it's important to look at the concept of chemical space. So chemical space refers to a theoretically possible number of molecules that can exist in the world. So they, don't ask me how these organic chemists came up with this rule, but basically they posit that, molecules up to 13 atoms, there are 977 million possible molecules that could exist theoretically in the world. If you go up to 17 atoms, that number goes up to hundreds of billions. And then drug space can occupy up to 40 atoms. So that's roughly up to 10 to 60 molecules. That number is called novemdecillion. Now, the database we talked about earlier, Campbell only has 2.2. So you see, we only have a very small sliver of the entire chemical space that we're not really testing. So very basics of basic machine learning and how it can help navigate that chemical space. It's basically machine learning is giving computers ability to learn without being explicitly programmed. So if you imagine tic-tac-toe, there's probably a limited number of scenarios you could code for, and it's humanly possible to code for every scenario until programmed to put X's and O's here and there, depending on what you see. But as you increase in complexity, like chess and like AlphaGo, you need to have a lot of training data sets from previous matches and have the machine learn to predict the next move based on its experiences. So there's three major categories of machine learning. There's supervised learning, which mainly is good for classification regression problems. unsupervised learning, which is good for clustering problems, and reinforcement learning, which is for goal-oriented tasks. So if you see, self-driving cars, or on YouTube, you watch, computers playing Mario Kart or something like that, that's reinforcement learning. And a subsection of that is called active machine learning, and that's what's used in pharmaceutical development. So the goal of active machine learning is to train the algorithm and then have the algorithm tell you which experiment to run to improve the model. So it's a constant feedback loop. So let me just go to this chart here. So you can see you start out with compounds, not known activities, you train, and then the model is trained. Now it can make some hypothesis. And there's two ways for this model to make a prediction. One is you ask the model to give me the best molecule that will bind to the receptor, right? So that's called exploitative strategy. And the other half is explorative strategy, where you ask the model, give me the molecule that you want me to test for, because you lack knowledge in this area, and I can feed back the experimental data back into the training set to improve the model. Obviously, you kind of have to thread the needle. You do want the machine learning algorithm to give you, good molecule to test, but you also don't want it to just keep giving you the best one, so it biases itself over time. So you want to balance up the two strategies. So this is a paper that came out, I think, 2 years ago, where they used this method to discover a novel antibiotic. So the motivation for this study was that they cite growing antibiotic resistance. You probably get calls from ID all the time about this, decreasing development of new antibiotics, and prediction of increasing death attributable to resistant infections up to 10 million a year by 2050. Now, antibiotics has been one of the fields of pharmacology that's been resistant to high throughput screening for whatever reason. And prior antibiotics were discovered through getting soil based microbe for metabolites and looking at which ones sort of prevented pathogenic bacteria. And then once you have a candidate antibiotics, you would sort of change the minor structures of that to improve potency and decrease toxicity. So, we talk about chemical space, we talk about lack of, breakthroughs using high throughput screening. So this is a prime target for AI to come in and try to solve this problem. So what they did is their training data consists of about 2,000 compounds with various levels of inhibition against E. coli. So they trained this model with 2,000 compounds, and this model is able to predict the probability that the new compound you ask to predict, it will predict how much it will inhibit E. coli based on its structure. So the model they feed in includes their chemical structure, their lipophilicity, their weight, and all these things. And then this model is now able to tell you, given the new structure of this molecule, will it inhibit E. coli growth? And they can now apply this model to multiple chemical libraries spanning 107 million molecules. This is way bigger than what they could do with high throughput screening. And they were able to select 99 molecules that were most strongly predicted, and they were tested in vitro, right? So there's two graphs here that I want to go over. In figure D, these are 99 molecules that were predicted to be highest in inhibition of E. coli. And 51% of them had no growth. So on the y-axis is OD. OD stands for optical density. So basically this is an analog for how many colonies there are. So lower the number, more inhibition you got. So 50% of the 99 predictions were very good at inhibiting E. coli growth. They also pulled out 63 molecules from the model, which had the lowest inhibition, and they tested these in vitro as well. And only 3.2% had the inhibition of E. coli. So it's pretty good at predicting which ones would be inhibitory and which ones are not in vitro. And out of the 51 of the 99 they screened that were actually inhibitory, they try to look for compound with low toxicity and that were most structurally different from the training set. and identified this molecule called halicin that actually was a preclinical drug under investigation for treatment of diabetes. So this is not a molecule that they would have stumbled upon normally. This is very different structure than normal antibiotics, but they found this. And you can see that as you increase the concentration of halicin here, OD or the colony growth goes way down. So it's very effective at inhibiting E. coli growth. And they also found that this is a broad spectrum antibiotic. I think you can ignore the black dots there. They just ran 2 sets of experiments. They just averaged that out. But on the bottom is various genes associated with different multi-drug resistance. And I think leftmost one is sort of the wild type E. coli. You can see there's some ampicillin resistance and MCR resistant. And with the halicin MIC is the minimum inhibitory concentration. So at normal concentration, it is able to inhibit E. coli with various different resistant genes inserted into it. So they posit that how could this be? And their theory is that this probably acts in a novel manner that's the bacteria is not used to seeing. And so they picked 3 organisms that's classified by WHO as the ones needing the new antibiotic therapy the most. The first one is the carbapenem-resistant Enterobacter, CRE, Acinetobacter, and Pseudomonas. And they ran a broad-spectrum antibiotic panel. And you can see at pretty low concentrations, it is able to inhibit the first 2, CRE and Acinetobacter. Pseudomonas, they had to reach very high concentration to do that. So at that point, you call that ineffective at that point. So 2 out of the 3, it could inhibit that. So that's pretty amazing. And they also tested against multi-drug resistant tuberculosis, and it is effective. And you can see with no drug versus halicin, over time, you see the colony forming units going way down. So this is a very effective medication against multiple different pathogens. They also conducted animal studies. So first study, they created a skin wound in mice and infected the skin wound with Actinobacter infection. And they treated with a gel containing no drug versus halicin. And I think after 72 hours, there's no colonies almost in the halicin arm. And then in figures E and F, they gave mice ampicillin to clean out their gut microbiome, sort of to reset. And they infected their gut with C. difficile, a very resistant organism. And they treated them with either vehicle, metronidazole, standard practice, and then halicin. And you can see over several time points here, 24. So after 72 hours, you start to see blue dots going way down in their fecal C. difficile count. So this is also very effective at clearing out C. difficile infection. So this study kind of blew my mind at how effective this could be. But how do we apply that to neurology? I think there are some challenges that are not there with antibiotics. Antibiotics can quickly be screened using machine learning, and candidate molecules can be tested using very quantified markers like OD600 in vitro. But I can't really think of a good correlate in neurology where we can do this quickly in vitro. We have biomarkers like amyloid beta and tau, but it is sort of debatable whether they're pathogenic or sort of side product of underlying disease processes. So we're going to need a lot of effort into novel target identification and biomarkers to test to similarly in this regard. So take-home points for my talk here. Drug development goes way beyond just clinical trials. It is time-consuming and costly. R&D is not the only factor in determining drug costs, but hopefully reducing this with more smarter AI, machine learning algorithms that will help you figure out, navigate the bigger chemical space can help decrease drug pricing, hopefully in the future. All right. Okay. So I want to thank my wife and my kids, my co-residents and colleagues, and all the faculty who taught me so much during residency. Ready for questions?
Thank you. Fire away, any questions in the room? Let's see if we got any on the internet. So how much relies on structure? Like just knowing with antibiotics, you figure you know what the target you're trying to attack in a bacteria. Can you do it just by matching drug shapes to targets on proteins rather than just doing this AI stuff?
I think that has been sort of exhausted. They've have beta lactams, and they've sort of tried to change structures of it to increase potency and decrease toxicity. So I think there are targets that we don't even know about that may be effective that can be discovered this way. So Hallicin was sort of an example of that. Yeah.
But I mean, there was a lot of press 6 months ago, some company that had come up with the structure of literally every protein that exists. And they said in the reports that this would enable drugs to be developed much easier. Is that a true thing, or?
I think it's optimistic. Even though Hallicin paper I thought was amazing, even with the 99 predictions they had, only half of them were really effective in vitro. So I can't say this is going to solve the problem. It will make it more effective, but I think there's still going to be problems that we need to test in real life. Yeah.
And my understanding is the pricing of drugs that are used in hospitals is governed by quite a different process than drugs that are used as outpatients, which is why there isn't a lot of incentive for companies to make antibiotics to highly resistant bacteria, because that's mostly a in-hospital kind of thing, where the rules of reimbursement are much less lucrative.
Sort of lump sum kind of thing.
Exactly. Yeah. That's my understanding.
I have a question. What's the magnitude of difference in the hits that AI found compared to high throughput?
For like antibiotics? I don't know the numbers of that. I haven't looked it up, so I don't know.
I'm just wondering.
Yeah.
So when penicillin was first discovered, it killed everything. Staph aureus, everything. And it didn't take long for them all to become resistant to penicillin. I don't know if penicillin kills anything. I mean, I honestly don't know if there's anything left that it works on. But yeah, they become very resistant. Anything on the internet? Hi, folks. We have a couple more minutes. Okay. 10 more. Yep. Kirby, you want to make comments? So Kirby was involved with drug discovery at Glaxo for quite some time.
Yeah, I found the talk very interesting. One thing that you're missing is that a lot of drugs still today are discovered based on biology insights. The SGLT2 inhibitors for diabetes, for instance, that was not the product of screening. That was the product of Ralph DeFrance's insight that peeing out the glucose would be good for the body. And it took him a long time to convince the pharmaceutical companies. When he did, everybody got on board, and there are multiple compounds. But that was a product of insight. And the GLP-1 inhibitors that we're all reading about for obesity is also the product of
Kirby and/or taking.
And/or taking. Yeah, there you go. It's also the product of biological insights. And another point I'd like to make is that using these two drugs, the two drug classes, many of the beneficial effects can't be predicted. For instance, when we got into inhibiting the kidney glucose transporter, we only thought we're going to dump glucose, and that's going to be great for diabetics. We had no idea about the beneficial effects to the heart that would be a consequence for that. and also the GLP-1 inhibitor or the agonist. It looks like, at least in mouse models, they have a great effect on AD-related neuropathology. Well, a lot of drugs do, so that may not mean anything, but it's promising. And I know that Novo Nordisk is pursuing some clinical trials along that area. And then going back to what Rich commented about Google identifying all the structures of all the proteins. And that will help drug discovery. The problem is that lots of small molecules find unexpected places to hide in protein folds and have unanticipated consequences. So even if you know the structure of the molecule, the small molecule, and you know the structure of the protein, you're not guaranteed of understanding what the long-term effect of the interaction between the two is going to be. It's a fun game, and it'd be great if everybody, could participate in it for at least a little while to really increase their understanding of how we can improve health.
Thank you for the information.
And then Kirby, there's just still so much serendipity, right?
Oh yeah, I mean, it's just a lot of it is just plain dumb luck.
Yeah.
For instance, the sulfa drugs. I mean, there are so many compounds that are spin-offs of the original sulfa drugs, and it just happens to be a great pharmacophore that hits a lot of different targets. And no one could have predicted that either when they were first discovered way back in what, 20s, 30s.
And how many Alexander Flemings would have come back from that vacation and looked in his petri dish and just said shit and thrown it out, right? You know what I mean? I mean, serendipity happens, but you have to appreciate it. I mean, I say this all the time. Everyone in your career are going to get 2 or 3 bits of serendipity. And the question is, do you throw it in the trash or do you recognize what it is? It happens to everyone. All right. Well, anyway, great job.
Thank you.
Thank you.